Hello and welcome everyone to this live event brought to you by Partners in Research Canada. My name is Stacey Joyce and I'll be your host. Remember that you can ask questions anytime during the presentation using the chat function in Zoom. Make sure you find that chat button in the top left-hand corner, and from there choose everyone to send your chat message to as opposed to all panelists. That way our audience members can see what other audience members are asking. Make sure to give us a shout out. Let us know where you're from, maybe what grade your students are, and if a particular student is asking the question and you wish to share, we'd love to know their first name so we can give them an individual shout out. So today's guest is Dr. Ralph Pantophlet. He is an associate professor at the Faculty of Health Sciences at Simon Fraser University in British Columbia, and he is the Michael Schmidt Foundation for Health Research Scholar there. Thank you so much for joining us. I'll let you take it away.
Thank you very much, Stacey, and welcome everyone. I'm trying to get my video to work, but it seems to be freezing up every time. So what I think I'm going to do is try to share some slides with you and talk a little bit about HIV. Talk a little bit about drug therapy for HIV, given that today is December 1st, World AIDS Day, and talk at the end a little bit about what the problems are in terms of developing an effective HIV vaccine, and some of the work that we're doing here in my lab at Simon Fraser University, and then I'll take your questions. So let me see if this is going to work. 1 second, please. Oops.
There we go.
So, okay. So hopefully now you're seeing a title slide that says Per Live Event, World AIDS Day, December 1st. So in case you didn't catch my name, Ralph Pantophlet, Associate Professor in the Faculty of Health Sciences at Simon Fraser University. So as I said, I'll talk a little bit about HIV in general. So on this slide, which I took from UNAIDS website, you'll see that in 2010, if you look at the top line, there were about just over 30 million estimated people infected with HIV. And last year, there were slightly more, about 37 million people infected with HIV. So despite a lot of progress in the field, there are quite a few people still globally infected or living with HIV, and this concerns all ages, so children as well as adults. On the right hand side, you'll see numbers for new HIV infections in 2010 and in 2000 compared to 2015, and you'll see that the numbers are still very, very similar. And you'll also see that not only does Africa continue to be the sort of the center of the HIV epidemic worldwide, with 19 million people living with HIV there, or over 20 million if you take both Eastern and Western Africa. And in terms of new infections, there is hardly a decrease in the number of people who are contracting HIV in those countries. Most alarming, I would point out, is that in Eastern Europe, numbers have actually gone up in terms of new infections, from 120,000 roughly in 2010 to nearly 200,000 in Eastern European countries in 2015. So if even though here in Canada there is a sense perhaps that HIV is a disease, or AIDS is a disease that can be managed. And that is certainly true. The pandemic is still raging on in other parts of the world, and so it is important. It remains an important problem. This graph shows you some of the effort to bring people who are living with HIV/AIDS, to bring them therapy. So the blue graph, the line, shows you that since the early 2000s, there has been a reduction in the number of people who are dying from AIDS-related causes, which is obviously a good thing. And at the same time, those blue bars that you see going from left to right are the number of people who are receiving therapy worldwide. So more people compared to 15 years ago or 16 years ago are able to access antiretroviral therapy, which is a good thing. It's about 2.5 million people across the world, so a tenth or less percentage of people who are living with HIV/AIDS. So it's not a lot of people, even though the number itself, the absolute number is great. But it's an improvement over what it used to be. It is certainly an improvement over what things looked like in 1981. So on this slide, you see some of the first news reports on when HIV and AIDS basically came on the scene, if you will, and this is from the US. So obviously HIV did not originate in the US. It originated in Africa and made its way to North America via the Caribbean, and basically popped up in places like New York and California, which is what this news report is about. And as you can see from the headline, they weren't actually talking about HIV and AIDS at the time. They were talking about people who were developing this rare cancer, and particularly in communities where you have men who have sex with men and things like that. And so they were talking about cancers, which I'll show you a picture of in a second, or other types of infections that normally would not afflict healthy individuals. And from a clinical standpoint, they were also reporting that people were losing very important immune cells, so-called CD4 T cells, and that was a clinical sign of an infection. So those were sort of the early days, and obviously now things have changed. So this is what that cancer that is referred to on this slide is what a cancer looks like. Maybe some of you have seen similar pictures from other places or from a movie. It's called Kaposi sarcoma, KS. It is a herpes virus, as indicated below the image. And characteristic for this cancer are these lesions on the skin that look like these rashes. Well, obviously what happens, it doesn't look very nice. So people were basically showing up at the clinic with these signs. And this particular form of this disease in HIV-infected individuals is a very aggressive form of the virus, of the infection. Nowadays, with antiretroviral therapy, we don't see this anymore because the ability of this herpes virus to cause infection is severely reduced as a result of therapy for HIV. So people are basically healthier and can fight off this type of infection by this virus. But at the time, back in the 1980s, this was not the case. So speaking of, I'll talk a little bit about retroviral therapy for a second. This slide shows you 3 graphs going from 1987, so the early days of the epidemic, to 1997, so a 10-year span. And it's talking about the efficacy of antiretroviral therapy for HIV, obviously. So in the early days, 1987-ish, there was one drug to treat HIV, and it was called AZT. And it's what's called a monotherapy because it's a single drug. And the graph, that red graph on the left-hand side, shows how the amount of virus in the blood of people who are infected with HIV, how it drops when they are put on this drug. And so the graph goes down, and that's what is indicated by those negative numbers. And they're basically just relative numbers, so the numbers in and of themselves don't mean a lot. But basically the graph shows that there's an initial dip when people go on therapy, and but then after within a few weeks, so what is depicted here is a 24-week time span. Within a few weeks, there is what is called a rebound. So the amount of virus in the blood basically shoots back up as the virus develops resistance to this drug. Then in 1994, additional drug was developed. So you have a 2-drug therapy, and you can see that even though the reduction in the amount of virus once people go on therapy is greater compared to 1987, even there, within a few weeks, you have a rebound of virus. And then in 1997, more drugs were developed. And this has become what essentially is the current standard of care for people infected with HIV. So people get what is called combination therapy. Some people call it HAART, so HAART, highly active antiretroviral therapy. And what you can see is when you administer 3 or more drugs, not only is there a significant or substantial reduction in the amount of virus once these people go on drugs, but there is also or hardly any viral rebound. If anything, over time, the amount of virus in the blood becomes or goes down to nearly undetectable levels. This slide shows essentially the same type of information. But instead of showing you the amount of virus, it shows you the number or the percentage of people who go on, who progress to AIDS. So the black graphs over a period of time, months. To the left, if you have no therapy, you will see that the graph shoots up. So the black graph shows you that within just a short amount of time, people at the time without therapy can progress to AIDS. With monotherapy, there's a slight decrease in the rapidity with which people go on to develop AIDS, but it's still pretty rapid, so within a few months. But then if you look at the red graph, which is the triple therapy, which is basically what you have now, is that people essentially, very few people, less than 5% of people who are infected go on to develop AIDS. So that's basically, as I said, the standard of care. This is a very busy slide, and I won't get into it. It's just to illustrate how research has led to the development of several different drugs that target different steps in the virus life cycle. So the virus attaches to a cell, and once it gets into the cell, it integrates the HIV genome is integrated into the host cell genome. And those boxes that you see everywhere are to indicate different types of drugs that block the process at different stages. Not only at the stage of entry, but also at the stage of when the virus gets out of the cell. There are drugs that are able to block what is called the maturation of the virus. So before the virus becomes fully infectious, there are also drugs to block all of those steps. And so basically what you have to imagine is that when someone is on triple therapy, they're getting 3 drugs that target different parts of the virus life cycle, and that is simply to prevent the virus from developing resistance against any one single virus. Resistance still develops, but it's very difficult for the virus to do that when it's being shot at essentially from different sides. So what I hope to have shown you is that we have therapy currently. It is very effective, but it's also well recognized that despite all of these advances in therapeutic approaches, that the only way to really curb the spread of the virus, as I showed you from those early slides, is an effective vaccine. It will simply take too long and probably cost too much to really be able to put everyone across the world on therapy. It's possible to do that in Canada, perhaps, but in other places it's much more difficult. So how do we develop a vaccine? Well, first, just a word on how most viral vaccines work. So most viral vaccines work through the action of antibodies, as shown in this cartoon. So antibodies that bind to entities on the surface of the virus. Those are called the virus spikes, which you may have seen pictures of. And those antibodies basically coat the virus particle and prevent it from basically getting to the cell that it needs to infect. It's a very simple model, and but that's basically how it works. So how is it that we're having so much difficulty to develop these antibodies or to develop a vaccine? Well, this slide shows you the challenge. On the top, you see the diversity of flu, either within a time span of a year. So you can see there's very little to see for flu, and the amount of dots or branches that you see is representative of the amount of diversity within the particular virus. So for flu, there's very little diversity, whereas if you look at below, where you have HIV, you'll see that the branches are much, much larger. Particularly on the bottom right, you'll see that in the African country of the Congo, in 1997, you'll see a whole bunch of branches, which basically indicates that there's a lot of diversity. So so HIV is very diverse, which is something that you probably are well aware of. And so one has to consider how it's going to be possible to develop a vaccine for HIV that makes antibodies that can tackle all of these different viruses. So what the research field and the work that my lab does is concerned with is to identify on HIV sites, areas that are conserved between all of these different viruses. And this is also a busy slide, but it's basically to illustrate that the field has identified several regions on the HIV spike that are pretty well conserved across the different HIV strains. And so the idea now is that we can take this information and try to elicit antibodies that target these very conserved sites. And this is basically how it's done. So we look at HIV-infected individuals. About 20% of those infected develop antibodies that actually recognize many different strains, even though these antibodies do not protect the individual that develops these antibodies. We can isolate those antibodies from these individuals. And then we characterize these antibodies for their interaction with HIV. And the idea is that we can then, so we characterize the interaction, and that we can then develop what are called immunogens, so we can develop what are potential vaccines that will elicit these original antibodies. And then if everything goes well, we would combine those entities into a single formulation that would then become a vaccine. So just to end sort of some of the stuff that one particular area that my lab is working on, which is on looking at, so what has become clear over the last few years is that the HIV spike, which is this gray blob that you see here, carries on its surface sugar molecules. Here on the left, depicted in blue, or here on the right, depicted in this pinkish color. So these are sugar molecules that a virus carries because it comes out of our cells, and these sugar molecules are made by our cells. So the virus basically coats itself in these sugar molecules, and that makes it somewhat undetectable to our immune system. But researchers have discovered that because there are so many of these sugar molecules, that the immune system of at least some people can or is able to recognize these sugar molecules. So we're trying to figure out how can we make antibodies that recognize these sugars. And so a couple of years ago, we published a paper where we described a bacteria that carries a sugar molecule that is very similar to what is found on HIV. And that sugar molecule is shown here. So on the top is what is found on HIV, and highlighted in red is the part of the sugar molecule that is shared with the bacterial molecule, which is on the bottom. And basically what we're doing is trying to chemically design sugar molecules here on the far right that resemble the sugar molecules that are found on HIV, which is shown on the left. And basically what we're trying to do is to make a synthetic molecule that when we inject it into someone eventually, will make antibodies that will not only recognize our synthetic molecule, but will basically cross-recognize the sugar molecules that are found on HIV because they share a certain resemblance. So I'll stop there and take questions, perhaps.
Great. So just to remind everyone, in case you joined us a little late, that you can ask questions by clicking on the chat button in the top left-hand corner of your screen. And then just make sure you change your to field to everyone, and then we can all see what each student is asking. And don't hesitate, just put those in freely, and then I'll ask them one at a time. So the first question I wanted to ask was, are there any theories why the incidence of new infections is not decreasing in Africa? So, our therapies are getting better for treating HIV. We're working on vaccines for HIV, but in the meantime, you showed a graph or a table that said that the number of new cases is fairly consistent. Are there any theories as to why?
I think it's, I've frozen up again, sorry. It probably has to do with several factors. One is that particularly in African countries, there continues to be stigma around HIV/AIDS. So, so either on the side of infected males who do not communicate to their spouses or others that they're actually HIV infected, because doing that would basically out them as being infected. There is also on the women's side who may have different sexual partners that they don't wish to communicate the fact that they are HIV infected. They don't want to, and communicate, I mean, by asking their partners to wear condoms, for example, because that might suggest that, the partner would ask, well, why do you need me to wear a condom? Because, you should be healthy and fine. And so, so I think there's a lot of stigma. And then perhaps third is that in many African countries, it's still, despite advances in therapy in and of themselves, many people still have a lot of difficulty actually accessing that therapy. So, so overall, I think that combination is why you actually, in the long run, do not, and on a global scale, you do not see a significant reduction. It goes down by 100,000 or so, but it does not go down to, or at least so far, has not gone down significantly.
Okay. We've got a question here asking, what inspired you to research HIV and AIDS?
That's a good question. I did not come to this field sort of in a direct way. I would answer the question by saying, actually, what inspired me to get involved in science was because I've always been interested from an early age in sort of the thinking about problem solving. And I was actually very poor at biology back in the day, so to speak. I was much better at chemistry and physics and those kind of things. But it was something about biology, and maybe it was the way that it was taught to me, where we always were asked to think about problems in biology. And so it always stuck to me. So when it was time to decide what I wanted to sort of study, go to university, I was very interested in microbiology. And I was, at the time, very interested in biotechnology. So modifying microbes to make, synthetic materials for food production and so on. But because of I was doing microbiology, I got interested in infectious diseases. So early on, I switched my focus from biotechnology to infectious diseases. And from some of the work that I did as a student, I became interested, in fact, in antibodies, and antibodies then led to becoming more interested in HIV.
Well, it's nice to hear that it's not necessarily a direct career path. I think a lot of students now aren't really sure what they want to do, but they sort of will pursue one subject or one subject area because they enjoy it. And it's nice to know that they don't have to have all the answers yet. They can go in that trajectory, and something may spark them to change direction again a little bit, and that's okay. And that's, I think, from doing these webinars, I find that's the most common trajectory, is a very zigzag type direction to a career path.
Yeah.
You touched a little bit in your slides on influenza versus HIV, and you were, I think, trying to highlight the complexity and the variance in how many different types of virus there are. Are there any other things that you can flag as differences between a retrovirus like HIV and a virus like influenza?
Well, from a biological standpoint, they are very different. So HIV is called a retrovirus because its genome, so the nucleic material that it carries inside it, is something called RNA. And it's difficult to explain what RNA is, but important to know is that RNA that the virus carries cannot be read, if you will, by the machinery of our cells to make viral proteins. The machinery, the way the cell machinery works is it reads DNA, not RNA. So the RNA of the virus needs to be converted to DNA. And the virus has an enzyme called a reverse transcriptase, a very long word, but it uses that enzyme to convert its RNA into DNA. And once it does that, it is that DNA that then integrates itself into the nucleus of our cells, the cell that is infected, and then basically the virus basically takes over that system, and our cell machinery then produces the viral proteins that it then needs to make a new virus. Flu is a different, different in that sense, that it is not a retrovirus. It is, it's a different kind of virus. It does not integrate its genome into our host cell. It does, however, need a cell to replicate itself. So in that sense, in terms of viruses, they are the same. But the way they go about making progeny, how they go about making new virus particles, is very, 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 very different.
Okay, very interesting. So the retroviruses can actually use that enzyme to create DNA within ourselves and sort of insert that, versus a virus that just uses us almost as ourselves as a host for making more of itself. Is that?
Yeah, yes. I mean, they both use our cells, but the way they go about doing it is very different. Another part that is different is the way that they actually get into our cells. So because HIV has this somewhat different genome, the way the whole process of once it attaches to the cells and once its genome gets into the cells, there are many steps for it to actually, molecular steps that are required for it to actually go from having this RNA molecule, which is the HIV genome, into DNA. Whereas with flu, it is there are somewhat fewer steps, and the whole process of how the virus that then gets put together inside the cell is different.
Okay.
Yeah.
Well, we're unfortunately running out of time here, so I just wanted to ask one more question, in that if students are interested in getting into this type of research, whether it's with AIDS or other viruses, et cetera, do you have any advice for them for how they can set themselves up for that in the most successful way?
Well, I mean, my advice is always that students should research the field that they're interested in, be it HIV or something else, and learn about the research that is going on. And as I said early on, I did not start off in this field early on. I started in a completely different way. So I try to use the stuff that I was learning over the years, try to apply it to HIV. So I think my advice to students would be to research what's going on, try to learn about different aspects, and even though it may not be something that you think might be useful, it is still good to dabble in molecular biology or biochemistry. And I mean, there's some basic things that are applied all the time, and you can use that information then later on to sort of specialize in a particular field, and you'll probably be set up very well in that regard.
Excellent. Well, thank you so much for that advice and for all the information today about HIV and where we've been and how far we've come and how much work there is left to do still. So thank you so much for your time and for answering our questions, Dr. Pantophlet.
Yeah, thank you very much, and it was my pleasure. And I'm terribly sorry that the video keeps freezing up. I try to turn it on every few seconds, but yeah, it just doesn't want to play along today.
Yeah.
But anyways, yeah, thank you very much. And if anyone has questions, I'm sure I'll hear them, or you can contact me directly.
Absolutely. Excellent. I will do that. Next week on PIR Live Event, we're presenting multiple Hour of Code webinars for all ages to celebrate Computer Science Education Week. More information on those webinars and other upcoming PIR Live Event webinars can be found at pirweb.org. So thanks everyone for tuning in, and have a great day.